Okay, let's take a look at our diagram associated with the cell. In this class, in prepping for tomorrow, Wednesday, we'll talk about a few of the components that we mentioned in our in-class lecture. And remember, when we're going through this information, copy or write down the things that I write down. We're trying to indicate what that specific location is and then a function of that location. So make sure you pay attention. I know the space is somewhat limited, so make sure you write neatly, and this will be a great study tool for our next quiz. Okay, our first component that we talked about, and you'll see on this diagram, is the smooth ER. And in our lecture we said the smooth ER has a couple unique components. We know, for example, it's a spot where cholesterol is synthesized, cholesterol is decomposed. We also know this is a location where there are no ribosomes. Okay, so we talked about smooth ER. Moving on over to the rough ER, we know that that location is unique as well. We know within the rough ER it has what we call fixed ribosomes, meaning that those ribosomes don't move, they stay in one place. We know that ribosomes are protein-based organelles. They synthesize those proteins, are pretty important in that development. And we know that within the rough ER, the building material for the cellular membrane is also produced. Okay, taking a look at our ribosomes. We know that ribosomes in this location are a site of protein synthesis. We know that there's two types of ribosomes, those that are free meaning freely floating within the cytoplasm. We also know that there are fixed ribosomes, and these are the ones that are studded on the rough endoplasmic reticulum. We gave this example in relation to that hard candy found on that wax paper in our discussion in class. Okay, the next component that we talked about in class, that we will for sure for Wednesday's class. I know we didn't get there in both, but the first being the Golgi apparatus. We know this is a pretty important component within the cell. It has a unique function. It modifies and packages the protein sent to it by the endoplasmic reticulum. So it receives those packages of proteins, and it does that in three specific ways. We'll talk about that process in our next diagram. So we know the endoplasmic reticulum is ribosome specific, but it also has another function. The ER sends proteins to the Golgi apparatus through transport vesicles or tubules. This entire process involves proteins being formed and packaged and exported to the Golgi apparatus. It all starts with the protein, right? We know the ER is protein specific. Ribosomes aid in that process. In this relationship, you can see the protein is modified to meet the specific need. An example of this might include attaching a carbohydrate to the protein to form glycoprotein. So once again, at this point, you can see the transition between 2 and 3. The protein is modified in order to meet a specific need. Once again, that protein enters a fluid-filled tubule or vesicle. This component pinches off of the surface to create that individual carrier. Eventually that vesicle or tubule makes its way to the Golgi apparatus and delivers the combination of proteins or glycoprotein that are required. Okay, obviously there's a lot of things happening on this diagram, so let's go through them one by one. We know that the Golgi apparatus that receives the packages sent to it from the endoplasmic reticulum. So it receives those packages. And you remember those packages were filled with a variety of different components, things like proteins, glycoproteins, things along those lines. Well, the Golgi apparatus continues to modify those packages. It modifies them in a way where they're able to leave the cell through exocytosis. And this is something that we'll talk about down the road. But three main pathways exist that allow this to occur. So let's go over some basic components of this diagram. At the top here you see the rough ER. We've already talked about that. We've seen the process through which those proteins and other additional materials moved from the rough ER to the Golgi apparatus via transport vesicles. You can see here we've identified this component as the Golgi apparatus as well. Pathway 1 is kind of unique. The first type of pathway involves secretory vesicles. These contain secretions that are discharged from the cell, and this allows these secretions allows the proteins to move to the environment outside of the cell. So through that process, secretory vesicles contain secretions that are discharged from the cell. This is one way in which those proteins are moved. Path 2 involves cell membrane components and vesicles. You can see this path, the same thing exists. Those vesicles move proteins to the cell membrane, allowing those proteins to interact with the membrane. We know from our previous section that that is an important component of cellular membranes, the addition of proteins. This type of path also aids in removing or recycling old proteins as well. The third pathway involves the formation of lysosomes and various vesicles that relate to that and promote that. So the Golgi apparatus produces vesicles containing digestive enzymes that become lysosomes. And as we know, those lysosomes are involved in breaking down waste, removing substances, and allowing them to be broken down in a way where they can be utilized and released by the cell. So obviously this was a lot of information, a lot of things to write down. Take your time, go through it. We'll once again cover this tomorrow in class. If you have any questions, bring those to class and we'll go from there. Thank you.